All right, good afternoon. Welcome, everybody. For those of you who don't know me, I'm Emily Ho. I'm a faculty in nutrition in the College of Health. Welcome to the College of Health monthly. I'm really excited for we can welcome Matt Norman Hord back to us here at OSU. A little bit about Norm, for those of you who may be more recent generation. Norm is a long-term colleague, friend. He spent 8 years at Oregon State in our college as our formerly known as Biopop school when it was that configuration. So have a leadership role, friends, colleague, champion of the college. Although he has not changed his letters or his colors in terms of his new institution, he's currently the department head of nutritional sciences at Oklahoma State University. So still OSU, still orange and black. A little bit about him before our OSU and after our OSU. He trained at several places, bachelor's at Michigan State University, master's at Clemson University, doctorate at Purdue, and an MPH at Johns Hopkins. He spent quite a bit of time at Michigan State University as a professor there before we recruited here to Oregon State University, where again he spent 8 years with us. Unfortunately for us, he moved on to become head of at the University of Oklahoma, a health sciences center, and then discovered he really missed the land grant myth of things and wearing orange and black apparently. So
I had it all anyway.
So has been since 2003, fairly recently, the professor and department head at Oklahoma State. He is one of the world's leaders in dietary nitrates and nitrites. Currently has funding through the USDA and the Office of Dietary Supplements to look at dietary nitrates in foods and contribute to the databases that are essential for us to understand food components and health. And I'll let Norm take the stage.
Thank you, Emily, for that introduction. It's great to be back. It's great to see friends and colleagues. Sean, can you hear me all right back there? Great. As Emily said, I've been studying this area for quite a while, since I was at Michigan State University, and I'll spend some time talking today about what I call a conundrum about dietary nitrate and nitrite. In other words, it's associated with certain health risks. And when I learned of this topic, I went to a seminar, a guy that was going to talk about health benefits, and boy, was I going to give it to him. And by the end of that seminar, I was won over and became a colleague of that individual and a collaborator, and now study those health benefits myself, and did so here at Oregon State University with Robin Tangway and others at the Thorough facility. I'm going to talk about that conundrum and then go on and talk about plant food origin nitrate and its potential as a conditionally essential nutrient that we have nominated it as, and then talk a little bit more about the biological functions and health effects of these nitrogen oxides. Talk a little bit about the risks associated with nitrate and nitrite, and then future directions. So because of the associated risks with nitrate, specifically in processed meats and exposure in infants, I've got to spend a lot of time giving you context about this before I talk about potential health benefits. And I'll start by introducing where dietary nitrogen comes from. It comes from bacteria in the soil, as well as lightning, which deposits nitrate in the soil, where it becomes a source for amino acid production in plants after it's fixed or goes through the nitrogen cycle. So this nitrogen cycle is represented here and is the source of nitrate in our diet. Humans are exposed to nitrate through foods, through water, and through beverages. And regional intakes reflect quite a variety of intakes around the world, with the US and Western countries being closer to around 65 to 100 milligrams a day than Eastern countries such as China and Japan, much, much higher in nitrate in their diets. Exposure to nitrate comes primarily through vegetables. Many people associate nitrates with meats and processed meats, but most of our exposure comes from vegetables, with drinking water contributing somewhat less, and processed meats actually quite little. If we follow dietary recommendations to consume a diet rich in vegetables, such as the AICR or the USDA recommends, actually 90% of the nitrate that we consume would come from vegetables. Nitrite exposure comes primarily from processed meats because it's a legally mandated additive to processed meats for food safety reasons. But this conundrum that I'm speaking of exists because we add nitrite and vitamin C to processed meats. And when they are digested, they produce nitrosamines, which are putative mutagens and carcinogens. So this has been demonstrated very clearly by systematic reviews by the American Institute for Cancer Research to result in a small increase in gastrointestinal cancer risk. And a different population, namely infants less than 6 months of age, are at risk for methemoglobinemia if they're exposed to too much nitrate in water. That risk of methemoglobinemia was terrifying because methemoglobinemia is experienced by babies as asphyxia. And because of that, some deaths that occurred in the 1940s in the US, federal regulations were instituted immediately to limit the amount of nitrate in water, and the European Union has a very similar protection on nitrate in water. This first point here is about how much we should be eating via diet, and I want you to pay attention to this amount. This limit is 3.7 milligrams nitrate per kilogram per day, which for a 60-kilogram adult would be about 222 milligrams. Keep that number in mind. This limit was set based upon one rat study that wasn't published, that was done in the early 50s. And we're still using that limit as guidance for these two inorganic ions. So this risk for methemoglobinemia, or so-called blue baby syndrome, was set because of these deaths. But many of us in the field don't believe that those deaths were caused by nitrate in the water for several reasons. Since then, infant dosing studies, dose escalation studies in infants have shown that high nitrate levels in those populations don't produce methemoglobinemia. Instead, we believe that the babies drank water that was bacterially infected, and those babies had gastroenteritis, and it was actually the nitric oxide and nitrate produced by immune cells that were activated in that infection that was really the cause of that methemoglobinemia. So folks in this field that I work in call this a hypothesis that's still in search of data. But it does drive policy still. I agree with the American Academy of Pediatrics statement on infant methemoglobinemia. In order to protect babies from contaminated well water, that the well water should be screened for nitrate, and that they shouldn't eat high nitrate vegetables for 3 months. So I'm in agreement with that and in support of it. I'm going to mention one slide about a big public health issue that's going on with regards to nitrite. It's not having to do with babies or GI cancer, but the fact that sodium nitrite can be purchased on the internet as a part of different suicide kits, and using sodium nitrite as a poisoning agent to commit suicide is very, very popular worldwide. So this is tragic. Those that use it have a mortality rate of about 30%. And folks are using this high dose nitrite ingestion as usually young people, teenagers, young adults, to produce this methemoglobinemia. asphyxia, and to produce death. So I gave a presentation at NIH a few months ago just on this topic due to their interest. So I won't talk anything more about it, but it's a topic of great concern. Turning the page to meat, we've used different sources of nitrate, like saltpeter, for thousands of years to preserve meat. This prevents the development of botulinum toxin in food, gives it a cured meat flavor, and helps inhibit warmed over flavor. Also helps maintain the nice color that we associate with meat. So the US FDA and USDA mandate that these be used in processed meat products along with ascorbic acid or erythorbic acid. And this is done to reduce the formation of nitrosamines in those foods to lower potential gastrointestinal cancer risk. Diagrammatically, this gastric nitrosamine formation is shown here, where dietary nitrate at a pH of 2 in the stomach and in the presence of vitamin C undergoes nitrosation chemistry to produce a variety of nitrosamines, which are mutagenic in their character and increase risk of potential gastrointestinal cancers. If you leave vitamin C out of processed meats, in experimental systems, we know that you actually don't produce these nitrosamines. So what's added to keep the food safe on the shelf produces risk in the stomach. This concern about cured and processed meats and gastrointestinal cancer risk has been well established by the American Institute of Cancer Research through a review of over 6,000 studies by 300 different scientists preparing these reviews for an estimate that colorectal cancer risk per every 50 gram per day intake is up about 21% in humans. What's interesting about this association is that the panelists who did this review could not determine a safe consumption level. In other words, there was a dose response, but not any intake at all is associated with increased risk of these foods. Again, a small increase in gastrointestinal cancer risk, but very consistent on many continents throughout the world. There are other conditions that processed meats are associated with, including chronic obstructive pulmonary disease and others. And this led the AICR to really give the strongest prescription for a diet that I've ever read, and that is limit the consumption of red meats and avoid processed meats. So we've got contexts where processed meats can produce risk. We have contexts where we've talked about infants being at risk for methemoglobinemia. I briefly want to mention that over-fertilization of agricultural lands can lead to eutrophication and high nitrate in water that's run off from that agricultural areas. And that particular type of exposure through drinking water is also associated with methemoglobinemia, colon cancer, thyroid disease, and neural tube defects in those that are exposed. And these levels can be actually quite low in drinking water. So it's very concerning that over-fertilization and other agricultural practices can produce drinking water contamination with nitrate that have risks associated with them. And I'm up here trying to talk to you about plant-based nitrate being a conditionally essential nutrient. Boy, do I have a hill to climb in the rest of this conversation. So clearly the food form that you're exposed to via nitrate is important, and the risks are differential. And this is due to food matrix effects and other things that we have yet to understand. I mentioned earlier, the WHO has this acceptable daily intake limit. It was about 222 milligrams for a 60 kilogram adult. I'm going to ask you whether that is that a maximum limit they're really talking about from those rat studies that weren't published? Or is it a minimum intake recommendation? And I say so because if you compute that acceptable daily intake limit for nitrate and nitrite, that 222 milligram amount in adults is exactly the amount in the first acute feeding studies that produced blood pressure lowering effects back in the early 2000s. So, so I'm not sure. My opinion is this intake limit is arbitrary, and clearly we can consume this amount and actually see potential health benefits such as blood pressure lowering. You might be asking, how much food does it take in the form of vegetables to get to 222 milligrams? And I took a picture of myself in my office because I didn't want to haul arugula from Oklahoma to Oregon. So I just took a picture. So that amount very, very easily achieved by diet, and early studies showed blood pressure lowering as a potential benefit. This idea of risk, differential risk, caused the European Food Standards Agency, which is the equivalent of our FDA, to study the matter. And in 2008, they concluded that exposure to nitrate from vegetables is unlikely to result in appreciable risk. And therefore, the beneficial effects of the consumption of these vegetables prevails. So how does this work? That dietary nitrate can produce these health effects. And now I'm going to go on and talk a little bit about what we know about the biology of how nitrate was discovered to become different nitrogen oxides in the body, including nitric oxide. Around the year 2006, this phenomena was discovered. In 1998, three gentlemen got the Nobel Prize for discovering this pathway by which nitric oxide synthase enzymes, which exist in three families, how they utilize arginine and oxygen to produce nitric oxide. And that was thought to be the primary, and is thought to be the primary source of nitric oxide in the body. But around 2006, we came to appreciate that dietary nitrate can serve as a substrate for bacteria on the tongue, bacterial reductases, to become reduced to nitrite, which can then in the stomach become nitric oxide. So the so-called human nitrate, nitrite, nitric oxide pathway was discovered then. And now we appreciate many different contexts in which the body, either on the tongue or within the body, can reduce nitrate to nitrite, nitrite to nitric oxide, serving not as the primary pathway for nitric oxide production, but a pathway that can rescue nitric oxide production in those physiological conditions that this particular pathway does not work, which is low oxygen conditions and in acidic conditions. We now know that the body spends lots of energy pulling nitrate out of blood plasma, concentrating it in saliva through this protein called the sialin family, solute load carrier protein. And in so doing, concentrates saliva at 10 to 20-fold higher than what's in plasma. And the nitrate serves as an electron acceptor during energy production in bacteria on the tongue. So it has a physiological function. And as those bacteria reduce that nitrate to nitrite, after a nitrate-rich meal, you can actually swallow millimolar concentrations of nitrite after a meal. We've now put together a story where we understand this oral bacteria have nitrate reductases. They reduce nitrate to nitrite, and then this nitrite is then absorbed, and then in the body becomes nitrite and nitric oxide, serving as another source for these important signaling molecules, nitrite and nitric oxide. One caveat here for this story is that we've done enough human studies now with men and women that we know that there's a sex difference. And we know that with the same dietary nitrate input in the diet and the same microbiome community structure on the tongue, women make more nitrite than men. Whether this is part of the average lower blood pressure observed in women isn't really known at this time, but could be an important factor in that biology. So we've come to know a lot about this, the microbiome in the mouth and in the tongue, on the tongue that achieves this nitrates to nitrite and nitrite to nitric oxide reduction. We know some of the families of the community of microbiome that do it. And we know some things that are now harmful, things like strong brands of mouthwash. Chlorhexidine containing mouthwash, which can effectively kill the bacteria on the tongue, can effectively ablate or block any blood pressure lowering from any nitrate source. Because again, those bacteria are required to reduce nitrate to nitrite. Grad Student Union presentation. Down bad. So again, chlorhexidine containing mouthwashes are blood pressure raising, and individuals who routinely use these on their mouth have higher blood pressures, and some studies are starting to show even increased stroke risk for knocking out those bacteria. Other potential benefits of nitrate-containing foods, plant-based nitrate-containing foods, include other benefits that are shown here for the mouth. So we're learning that these foods are not only good potentially for the cardiovascular system, but also for oral health. So we've proposed that dietary nitrate be considered for study as a conditionally essential nutrient. Just to remind everyone that in this country, we use a process hosted and run by the National Academies of Medicine to determine dietary reference intake limits for nutrients. There are several types of nutrient recommendations under the DRIs, all depending on the amount of evidence that's available for a specific nutrient. And these essential nutrients are established for healthy populations. Conditionally essential nutrients become indispensable under specific physiological conditions. And I'm going to describe some of those that relate to dietary nitrate. And so the National Academies of Medicine has amended their process for determining conditional essentiality to recognize that disease-related etiologies can change nutrient needs and physiological functions. Things like inflammation can impact absorption and lead to whole body or tissue-specific deficiencies. Neil-Ann, we started looking at this thing in dietetics about 2010, 2011, and started teaching this in our classes. We actually have guidance from our academy to do this. Now they're applying it to the study of nutrient requirements under those disease-specific conditions. So looking at atherosclerosis here, and as an example, nitric oxide becomes less bioavailable. Your vasculature produces less as you age and as you experience atherosclerosis, whether you have the condition of hypertension, chronic renal failure, or chronic heart disease, there's measurable decreases in production of nitric oxide by the vascular endothelium under these conditions. We now believe that dietary nitrate can serve as a substrate for nitric oxide production and rescue people from this deficiency caused by these diseases. So that's the basic proposition. And I said as you age or as you experience disease, but I want to make clear not every population around the world experiences this as they age. There are indigenous populations that have been studied all over the world. The Shamane and the Amundava in Bolivia and native tribes in Taiwan that do not experience increases in blood pressure as they age. They don't experience increased adiposity as they age. They're metabolically very normal as elderly people. So I'm going to say this is a feature of the sedentary lifestyles that we lead in this country and the Western world. The pathophysiological processes that are associated with atherosclerosis include endothelial dysfunction and oxidative stress. And these two processes are addressed through dietary nitrate consumption in human studies. So the process of determining whether nitrate could be a conditionally essential nutrient by the National Academies of Medicine would end up in a category called specialized nutrient requirements, that under these conditions of atherosclerosis, nitrate may be able to rescue the phenotypes of normal vascular function as a specialized nutrient requirement. So whether dietary nitrate is low or whether you're not making enough nitric oxides through the processes that I've just showed on the last couple of slides, that all of these ultimately result in low nitric oxide bioavailability. So this led us to propose, and this was back in 2009 that I first wrote it, that there's enough evidence for us to think about nitrate as conditionally essential. Nitrate-free diets in rodents lead to hyperglycemia, high blood pressure, adiposity, and premature death. Dietary nitrate and nitrite restore antihomeostasis in mouse models where the eNOS gene in the vasculature is knocked out. And this has been studied in that model as well as other animal models. And dietary nitrate and nitrite are undergoing study in a number of clinical trials supported by and listed on clinicaltrials.gov. My colleagues and I have done meta-regressions and meta-analyses in a recent paper to feature data associating dietary nitrate intake with positive disease outcomes, including incident hypertension, all-cause mortality, and cardiovascular risk, in addition to additional studies where we focused primarily on blood pressure. And in all of these studies, there's a consistent association with nitrate intake with positive effects on these cardiovascular outcomes. Beyond those studies, whether they were done in vivo or ex vivo, there's a great deal of basic science information in cells and culture and in many animal models that dietary nitrate affects these phenotypes that are listed on this slide. And so many of the things that we associate these positive benefits with dietary nitrate, nitrite, and nitric oxide, we can trace back to these physiological phenotypes that are associated with positive benefits on the cardiovascular system, including lower blood pressure and decreased platelet aggregation, increased glomerular filtration rate, and decreased risk of nephropathy, and better insulin sensitivity in tissues, and higher level of glucose clearance in skeletal muscle. So lots of reasons why the science base for nitrate and nitrite is strong, and anything in diet that produces these kinds of effects, you can bet will create a dietary supplement industry. And around the year 2009, the dietary supplement industry for nitrate was created. And they give these very rosy sounding advertisements for their product and its ability to improve nitric oxide in the body. But it's not just hucksters out there that are doing this. These are all my colleagues. I've published with each of these people. I have a paltry H index of 27. My scholarly H index is 27. And I think I've had about 4,000 citations of my papers. These people all have many multiples of my H index and my citations as medical doctors in cardiovascular medicine. Every single one of these individuals has started their own company selling or incorporated a company selling dietary nitrite products targeting the pathway that I'm talking about. And so I'm talking about really well-established, highly published, highly cited academics. Some of them are nitrate and nitrite sources. Some of these just add nitric oxide to oleic acid, which is rich in olive oil, and created a drug, this company called Complexa, which shows great efficacy against fatty liver disease and other end products. But these folks weren't the first to jump on it. This was published in 1909, where 80 milligrams of dietary nitrite was given. And these investigators back then observed this blood pressure lowering effect of nitrite. So we're only rediscovering something that back then, nitrite was a part of the therapeutic armamentarium of a physician. So around 1940, they stopped using it for reasons of not being able to get the dose right and perhaps some of the risk of giving too much. So I just want to say that at one point in time, this actually was a therapeutic agent regularly used by physicians. I haven't talked a lot about dietary nitrate and nitrite in terms of human performance. That's a whole field, as it's thought to improve endurance exercise and speed and power. We, in fact, with Dr. Laura Beaver in the back, did studies with zebrafish here at Oregon State, where we demonstrated some of these benefits in zebrafish to show that it's translatable even through that particular model. A committee appointed by the International Olympic Committee looked at the evidence about performance enhancement due to dietary nitrate and did in fact say that dietary nitrate does improve athletic performance in this publication. But there are limitations in our current science, and there's still a lot to be done. Back in 2014, I was a part of an NHLBI panel that looked at this. Seemed to be strong association between nitrate and lower cardiovascular disease risk. And we asked ourselves, what are our current limitations? One of them was that we needed improvements in standard databases to actually be able to express nitrate and nitrite concentrations in foods. Secondly, need to look more at the issue of drinking water nitrate content in order to better understand dose-response relationships in populations. We always say we need large cohort studies, obviously, to get the statistical power that we need to see whether the impact on physiological measures of dietary nitrate have strong veracity, and also to look at this issue of nitrate consumption and this nitrosamine formation, which has been an issue for quite a long time. I'm going to talk about two developments that have addressed one of these needs that we expressed back in 2016 in that publication. My colleagues at Edith Cowan University in Australia have done kind of a web scrape of all published data of nitrate, nitrite, and nitrosamines in foods globally, and through a very systematic process, assessed 129,000 nitrate values for over 800 foods in 80 countries. And then they are now, have now made available very recently a new database they're calling Nitr-Navigator. And this is a picture of this website and the folks that have done the work. So part one of this discussion is that these Australian investigators, using already published data, have created a database for researchers to use where we can look in a granular way at dietary nitrate intakes in a way that we haven't before. They have applied their database to a couple of different prospective epidemiologic studies. The first one here is the Danish Diet, Cancer, and Health study, and have found in that cohort study that systolic and diastolic blood pressure lowering was observed in those populations, and that nitrate intake was associated with a lower cardiovascular risk reduction. So this is the promise of having better data sources for epidemiologists to do this kind of work. They've applied it to an Australian diabetes, obesity, and lifestyle study, which is actually at the time one of the largest prospective studies done in Australia, and found lower all-cause mortality, lower cardiovascular mortality, and demonstrated clear dose response effects in that population. So they've been working on that project for over 15 years. They have published 5 papers on their database, the folks in Australia, and I'll credit Dr. Catherine Bondano for leading that effort. At the same time, we've been funded by the USDA and the NIH Office of Dietary Supplements to actually measure foods and express that data in a special interest database for the USDA. So this is the leader at USDA who's funded our project, and this is our lab at the University of Oklahoma Health Sciences Center, and my co-PI on this, Dr. Frank Hayes, along with 2 PhD students, Michelle, Anna, and a master's student, Diane Whitehead. It's very interesting doing this work. We're in our 3rd year of funding. We just secured a 4th year of funding for this project to measure nitrate and nitrite in food. And we get together monthly to be grilled, basted, and fried by these experienced investigators at NIH and USDA. So while I think what we're doing isn't rocket science, we're measuring nitrate and nitrite in thousands of foods. We have the collaboration of lots of folks at the NIH and USDA on a monthly basis for our work. It's a lot more difficult than just writing up a grant report, I'll say. Did I say micromanagement? Anyway, where do we go from here? There's a lot of things we could do to try to really try to move this conversation about dietary nitrate to the forefront. I really believe there's public health benefits from people consuming more leafy green and root vegetables, which are the richest sources of nitrate. We can go the dietary reference intake route, but that requires lots of money and a federal process to engage the right people to get that done. And there's lots of nutrients that are in the queue to be studied that way. So we've chosen to take another route, a recently created dietary guideline process through our professional society, the American Academy of Pediatrics. And so I'm working with several of my colleagues to come up with a guideline for plant-based sources of dietary nitrate. And this is based on another dietary guideline that had been produced, the first, on flavonols and cardiovascular benefit. Flavonols that you may associate with being rich in dark chocolate. So Christy here, Dr. Christy Curl-White, led that particular effort. And we're working with a cardiologist from London named Amrita, and 2 women that I actually was on their doctoral dissertation committees at Michigan State, which kind of dates me a little bit, but the chief science officer for the Academy and the chief systematic analyst for the Academy of Nutrition and Dietetics are on it, as well as an English colleague here, Oliver Shannon. So I'll conclude with sort of a summary slide of where we think the science is at for this particular set of dietary components, that plant foods and foods that contain nitrate and nitrite from so-called anthropogenic sources on the right are processed differently, metabolized differently, and result in different physiological outcomes based on this difference in metabolism. So these plant foods become these biologically active nitrogen oxides. They produce many different products that they're either nitric oxide, a nitrolipid, or nitrated lipid, or other proteins. And these are all associated with metabolic phenotypes of health, such as lower risk of hypertension, atherosclerosis, chronic kidney disease, and metabolic disease. Whereas these anthropogenic sources lead to gastric nitrosation reactions, as do these, but the components in these plant foods, vitamin C, flavonoids, fiber, have a matrix effect in which they actually don't affect tissues in a way that stimulates carcinogenesis or decrease endothelial function. So these products are then associated with increased risk of GI cancer and methemoglobinemia in infants. So just to conclude, I think that there's a large body of evidence that dietary nitrate from plant sources are vasoprotective, blood pressure lowering, and I didn't talk much, but they also block platelet aggregation very powerfully, which is another third powerful mechanism for cardiovascular protection. And I think we should engage a regulatory paradigm to examine whether or not these foods containing nitrate and nitrite could be considered conditionally essential nutrients. While this U.S. Institute of Medicine DRI process is the gold standard, probably not realistic for us to engage that process on our own. And we're going to continue to work through our Academy of Nutrition and Dietetics process to make this guideline to elevate this potentially conditionally essential nutrient for our researchers and for Americans who would wish to benefit from consumption of these foods. So with that in mind, I will conclude and thank you all for your attendance.
Online, but came over here to ask it in person.
Oh, gosh.
Hello
I'm just
there.
curious because
Good to see you.
earlier when you were talking about the processed meats versus like the vegetables, a lot of processed meats now, like the lunch meats, they're nitrate free. Are they safe? Is it like they're using a different process that might be just more dangerous, or what is your?
So food labeling laws in this country allow processed meat makers to use alternative sources of nitrate and nitrite instead of the sodium salt of nitrate and nitrite. So they can say nitrate free. But what they use is a dry vegetable powder rich in nitrate, usually celery powder, and they will culture bacteria with that powder, which helps it reduce nitrate to nitrite. So it essentially has nitrate and nitrite in it as if it had nitrate and nitrite salts. And every nitrate-free meat, and especially the big national brands that are on TV and saying nitrate-free on TV, we get more nitrate and nitrite in those products than we do in conventional processed meats. And it's been that way. We first measured it in 2008, and it's still those nitrate-free sources are still consistently double and triple the amount of nitrate and nitrite in those products.
I'm going to agree with Norm there because I was doing some of those measures and it was surprisingly high sometimes. I won't buy those. I'd much rather get the ones that say they have got nitrate in it because I'm like, I know that this is a controlled amount going in that food, or this other process where they think where it looks better. It was so variable when we were testing it in the lab, and sometimes very high.
Sir?
I have a couple of questions. First, when we eat cured meats with nitrate and nitrite, if we take some vitamin C with it, Tafer?
No, so there's a gastroenterologist in Glasgow, Scotland named Ken McCall, and he did the kind of experiments that we do in the lab where you leave one ingredient out of the mixture, and then he looks at nitrosamine formation in a sort of a simulated gastrointestinal system. And what Dr. McCall found was the most potent stimulator of gastric nitrosamine production in this mixture that you're talking about. You've got protein, fat, vitamin C, nitrate, nitrite. The most potent stimulator of nitrosation chemistry in the stomach was vitamin C. And if you left it out, you got very little nitrosamine formation. The problem is it's very effective. Vitamin C is very effective in the food, on the shelf, in the refrigerator, at keeping spoilage organisms down. So, would you sacrifice food safety for lower potential GI cancer risk? Yes.
This may be too early in your study, but have you noticed substantial variation of nitrite values in the same vegetable, two different sources? And for example, is organic going to be different?
Organics almost always going to be lower than something that's been fertilized. I showed you the picture of the Zoom call with all of our little postage stamp photos on it of our collaborating group at NIH and USDA. And I have to admit, now that my seminar is over, I have to admit I'm a pretty reluctant researcher of this topic in the sense that, measuring things in foods isn't going to get you a big NIH grant, even though in this case it did, and they keep chasing us and keep giving us more money. The USDA has stored foods for decades in big freezer farms they have in Beltsville, Maryland. And we have just received over 1,000 foods going back to the 1940s. And again, I don't think necessarily nitrite would be stable under freezer conditions that long, but nitrate probably is. So we're actually going to go back under the USDA's request, and we're going to go back and measure all these foods. And I use the word reluctant as a researcher because is it that interesting? Is this that interesting? But they're giving us money to do it, and we've got great folks in the lab. So we'll see whether over time nitrate and nitrite may have changed.
But even today, from different sources, have you noticed a noticeable difference?
Without a specific example, are you asking me about a specific food? I
Just
don't.
in general.
Yeah, I don't think I could make a general statement. Bar?
I'm just wondering on that summary slide that you left out the supplements, and I was kind of thinking supplements could fit on either side. So I was just curious where you're at on that literature right now. Do you think the supplements that people take for exercise fall more like foods, or is it more like the exogenous?
I got involved with the first company that started making dietary supplements, and my involvement with that company bridged somewhat my time here at Oregon State, and about a year or two into my time here, I quit. I wrote the product insert for those dietary supplement products. That was my job for the company. And what I said was, you should consume this supplement, and that supplement contained dietary nitrite. I didn't make a big deal of that, but actually, we got a whole nother year of funding for this project because NIH Office of Dietary Supplements wants us to do a ton of dietary supplements for this project. That product insert that I wrote said, consume this product 1 hour before or 2 hours after a meal. Why did I say that? Because I didn't want you taking this big honking 20 milligram dose of nitrite, which is as powerful as 1,000 milligrams of nitrate, with a meal, and then engage all this nitrosation chemistry with other meal components. So that's actually the first thing I did, and I thought that as a registered dietitian, as a health professional, I thought that was my job to protect the public.